All right, let's jump in. I think I am very much like all of you when I say that I am literally buried in work and have realized that I'm so far behind that I don't know if I'll ever catch up. So if you're feeling that same way, don't feel bad. I did receive a couple of emails from some of you this weekend just asking if there's any possible way to extend the deadline for the homework. Of course, we are at that point where I don't mind extending the deadline because we only have one more traditional homework assignment left. So towards those ends, we push that deadline back to this Friday at 2 p.m. Remember that we do have Open Lab from 12 to 2, so if you want to come to Open Lab and get help, you can. I will not be in Open Lab this week. I will be at the Tobin Spittlary Luncheon and will be hopefully writing my speeches for that the night before the luncheon. That's where we are right this time of year. It's always everything at the last minute. But I hope that as you receive feedback from me on your poster abstracts, that you're able to give those a little bit of TLC and a little bit of attention to turn those around. My objective is to create an abstract packet that, as I mentioned earlier, will be distributed to the faculty all around campus. And to anyone that you think you might want to come to your presentation, you can send them that abstract packet so that they know what to expect. They can get all the movie trailers for the main events at the presentations. So get those back to me when you receive feedback. I am working through them very slowly on my end. So as I do get to them in order that I received them, then hopefully you'll be getting back to me on them. Okay, I think that's most everything for lecture. Please keep working to submit the fever discussions and get those wrapped up and just come to an end with your discussion on that. The unknown report is due for lab, and that's another reason that it seems very prudent for us to push off the homework deadline. I don't want you to have to compromise one or the other, so please get the unknown report done and turned in. If you haven't used your late coupon yet, you are allowed to use your late coupon on the unknown report. That does buy you an extra 7 days a week, and then you can submit next Tuesday. Actually, it is of note that next Tuesday is our last day of lab. Melanie?
Do we lose our point for formatting if we opt to do the landscape for a table instead of
No, Yeah, no, do whatever for the tables that makes them less cumbersome, because they could potentially be very cumbersome. I've seen people turn in ones that you have to tape together multiple sheets in order to see the table. I'd much prefer that you make them readable and not and make them look good, as opposed to trying to make them very, very large to fit some sort of formatting requirement. The formatting requirement is really basically there for the main body of the text. So that's where we want it to be double spaced for reading, you know, to make reading a little bit easier. Tables are actually easier to read when they're in a smaller font and they all fit onto one page, as opposed to trying to make them double spaced. And I did get a couple questions about that. Anything else on your unknown reports, Julia? 8 is the minimum, and 12 is the maximum. No, fronts. Like, yeah, just fronts. So it is hard to fit it all into 12 pages, I know. I don't really care if, as I just said with the tables, if those are a smaller font. I mean, those actually probably should be. And that's the big bulk of it, right? I mean, the tables are what's taking up about 7 pages, right? Or something like that. It depends on how you formatted them. So, you know, just adjust the formats a little bit to allow that 12-page limitation to not be too restrictive. Sammy? You know what? That is 100% in your TA's jurisdiction. Yeah. So if your TAs have said that they don't mind getting online or they want it online, then by all means submit it on Wild Courses. If they'd prefer a hard copy, then try to get that to them too. A lot of them are working to adjust to the online grading forum, and some of them like it and some of them don't. So just check with your TA on that. Okay. Anything else? Okay. Today is both one of the days that I most look forward to all year, and it is also one of the hardest days for me. We'll be talking about HIV/AIDS, and I hope we'll be coming away today with a lot of important take-home messages, but also the inspiration to take the course that's offered on campus called HIV/AIDS that does last for a full semester, though it is commonly offered in the pre-session in summer. So it's a great class to take at the very beginning of the summer to kick your summer off with a lot of really intellectually rigorous material to really get you thinking throughout your summer break. So let's jump into our ending of what we were talking about last time. If you recall, when we left off last time, we had been talking about replication within the polio virus. We were using it as an example of one strategy of many that viruses might use in order to replicate their nucleic acids. We had ended right there and hadn't said much about maturation, though we aren't going to say much. That is, just like with the world of bacteriophages, maturation refers to the process of putting together the virus after it has made all of its parts. That is, packaging the nucleic acids into the capsid and making the fully formed daughter viruses. The daughter viruses, of course, then have to get out of the host cell. And there's a couple different strategies that they can use for getting out of the host cell. What was the strategy that the bacteriophage, the T-even bacteriophages used in E. coli to get out? Lysis, right? They just simply completely lysed the host cell, allowing up to 200 or more daughter viruses to escape at that point. Now, that is a strategy that is used by some of our animal viruses, human viruses, is to lyse the host cell. Oftentimes, this is due to weakening of the host cell during infection. That then, of course, releases the daughter viruses. Sometimes this is actually a plan gone wrong to a certain extent by the immune system. I don't know if you recall talking a little bit about the fact that every nucleated cell in our body actually flies a flag of self. So literally hoisted up on a flagpole called MHC, or Major Histocompatibility Complex, every one of our cells hoists up a flag that says this is the particular constituent within me, the protein constituents within me, and if it flies a self-protein on that so-called flagpole, then the immune system comes around and says, you're fine, you're fine, you're fine, you're fine, oh shoot, pirate flag. And if it sees a pirate flag, what I mean by that is that it has put onto its MHC protein, it has put a non-self protein, an antigen, meaning that the cell has been invaded by perhaps a virus. When the immune system sees that, it says one of two things to that self cell that is flying a pirate flag. It says, one, kill yourself, or two, I'm going to kill you. And sometimes it will literally release cytolytic toxins and kill the self cell gone wrong. But why does this backfire? Why might it say that sounded like a good plan but wasn't? You got it. It allows for lysis of the host cell, allowing many daughter viruses to get out. So it's a good plan to destruct that cell that has been taken over, except for when you realize, wow, that's releasing all of these daughter viruses, okay? So this sounds like something that could definitely be represented in some of the video games that Aaron and Zach's group are doing, right, for their poster presentation that we're looking forward to. Pretty dramatic, right? When these proteins of non-self are displayed on the surface of a cell, and they cause destruction by the immune system, sometimes that has the negative impact of allowing all the daughter viruses to get out and go and infect more host cells. Now, the really smooth criminals, they aren't the ones that are lysing and burning through their host cells. In fact, sometimes I think of these viruses that quickly and rapidly lyse their host cell as sort of being the stupid ones, because they're burning rapidly through their host cells and they're making themselves very known to the immune system. The much smoother criminals are those that leave by budding. So when a virus gains its envelope by budding off of the host cell, not only does that leave the host cell alive, but it also means that the virus takes what with it? That is, its envelope is what? Self, right? And that is so smooth, because it means that it leaves the virus looking like the host cell. It has immunoglobulins and other things on the surface of the cell potentially that say, hey, I belong here. So then the virus goes undetected. And over very long term, it makes the immune system so confused that with chronic infection, sometimes the immune system starts attacking itself, called autoimmune factor, right, effects of a chronic viral infection. So this is a very smooth strategy for leaving the cell. You can see here the nucleocapsid. Notice that the viral spikes are inserted into the host cell membrane, and then when budding takes place, not only does the virus take with it the spikes that are its own, but it also will potentially take with it, or does take with it, many of the integral membrane proteins of the host cell. So when it is released, it does, as all of you so thoughtfully said, look like self. So which one of these types of viruses is most likely to cause a chronic or a latent infection? That is one that's always in you. Yeah, envelope viruses. And that's a broad generalization, but it allows us to think about the fact that these envelope viruses that are leaving by budding might hang around. And in fact, it's interesting to note that the herpes viruses do something even slightly different. They bud off of the nucleus, so they actually have an envelope made of nuclear membrane, and then they're shipped out by the cell naturally by exocytosis. So it's another smooth way that they have self on them, but they just get it from a different membrane, the nuclear membrane, instead of the cytoplasmic membrane. So looking then at these characteristics of chronic versus latent versus acute infections, acute infections are kind of the stupid ones. That is, they're really rapid onset. We get symptoms right away. Lots of cell lysis is going on. For the most part, the host cells that are infected die. And the immune system rapidly recognizes and gets on top of the infection. So an acute infection, short-lived, localized, you get symptoms, but quickly your immune system responds, and you develop that memory. So you get long-lasting immunity against these infections, and you clear them rapidly. What would be a good example of this? Something that we get, and for about 7 days we're real sick, and then boom, we get it out of there and we're flu, right? The influenza virus, the rhinovirus, the common cold, right? These are things that are acute infections. They're quick, they're short-lived. We make good, long-lasting immune factors to respond to them upon a second exposure. Julia? I have a question. Yeah.
So like when you were saying about pirates and stuff or whatever,
yeah.
basically when you get sick, it's like the virus is winning for a short second until our cells take over and start to...
Yeah, I mean, and that's a really good point. Her question is, so, you know, the pirate flag thing, when a pirate flag, when an MHC molecule, it displays antigen on its surface, aka a pirate flag is flown, the virus is winning in that cell, right, in that one cell. But that doesn't necessarily mean that it's winning overall, right? It could be that that cell comes very quickly lysed, whether by immune or by weakening of the cell. And then, of course, eventually, you know, when you have the flu, there's a lot of localized cell death in your respiratory tissues. You know, there's a lot of cells fighting and dying in there. But you don't ultimately fall, right? So there's local losing, losing the battles, but winning the war, okay? So acute infections are very, very contrasting with persistent infections. And persistent infections are those that we always have, right? We all have viral infections. In fact, I just read a really fascinating article about the fact that about 8% of gene sequences in the human genome actually have retroviral roots, which is really interesting to think about, as we were talking about last time, evolutionary connotations of viral infection. So we all have these persistent infections, and we are all termed carriers. These infections don't ever go away. These include things like herpes virus. We can split these into two categories. There are those infections that, although they may become asymptomatic, are always present. We call those chronic infections. These include things like HIV and hepatitis. We're going to talk much more about HIV, but take note that no matter what, there's always some sort of detectable viral titer. That's very different from a latent infection in which they actually become undetectable. They're completely dormant. And this can be things like herpes. Our fever blisters, they only show themselves when we've been immunocompromised in some way. Varicella zoster, remember talking about that last time? It lays dormant in our neural dorsal root ganglia of our neural, like cranial neural cells, and it comes out when we get shingles, right, later after having had chickenpox. Epstein-Barr, that one causes mono, right? We talked a little bit about that last time. And I was just reading this morning, because we've been thinking that one of my athletes has been chronically sick for a very long time, and we think it might be that it's a reactivation of the Epstein-Barr virus. And I found a fascinating article today that talks about how Epstein-Barr reactivation seems to be more common in elite athletes. So that's an interesting thing to think about, is the reactivation of mono. So all of these are latent infections. So we understand them to be around, but not always detectable. The last category that I want to talk about could have been categorized under persistent infections. However, because it is poorly understood and because it is unique, I'm talking about it as a separate category. These are what we term slow infections. That is, in very rare cases, very unique responses to only some viruses. For example, there's one caused by the virus that causes measles. It causes these very slow onset infections that are only found after years after having had the original acute infection. And they manifest as neurodegenerative diseases. Now, most people, by far, most people who have had these viruses don't get these slow infections. But occasionally, every once in a while, someone who has had the measles virus will develop subacute sclerosing panencephalitis. It's a fatal and neurodegenerative disease. So it's very interesting to think honestly about the little that we really know about viral latency. And it's something that is a really active area of research, and maybe one that you'll want to dork out with a little more yourself. But I actually want to take a little bit of our time so that we can talk about HIV. I think that it is so very important, and perhaps because we have become used to it, it's something that no longer gets a lot of our attention. HIV really is the great pandemic, of course, of my lifetime, but I would argue also of your lifetime. 39 million people have died of HIV-related illness. So when we think about that number, 39 million, and we think about the fact that in 2012, 1.6 million people died of HIV-related illness. In 2013, 1.5 million people. And then we start to think about the news coverage that was dedicated to Ebola virus, and where is our current annual count? Who remembers? I challenged you last time. 10,000. I challenge you now to think about that, and to think about why. Why is it that we talk like crazy about something like Ebola, and our news is full of Ebola, And yet every year we're seeing 1.5, 1.6 million people dying of HIV, AIDS-related illnesses. Feel free to speak out at any moment if something hits you about that, but mostly I want to plant the seed, Nathan.
Part of it could certainly be due to the fact that Ebola is much more quick, faster acting. So
It's dramatic, right? It's very rapid to kill. Okay, good. Yeah, I think that could be part of it. Sammy? Yeah. Mm-hmm. Yeah. Yeah. You got it. You got it. HIV is the disease of the othered, right? When I say that term, I say it with a capital O, othered, right? Those who are stigmatized, those who are shamed, those who are punished. We associate HIV with those people, right? Those othered groups. And because of that, we want to talk about diseases that seem to hit those who are not othered. And I think that's something very important. And it's also a reason that I think it's very important that all of you are never hesitant to talk about HIV. And please fill my classroom, and anytime you ever want to talk to me about something othered, I love the othered. It's what I most identify with. So please give it voice in your lives and in your spaces. And let's talk a little bit about the microbiology of retroviruses. And sometimes I like to joke that people think that we call HIV a retrovirus because it's associated with the 80s. And in fact, I do have a little model of a retrovirus that looks a little bit like an 80s retrovirus. But in fact, that's actually not why we call them retroviruses. The reason that we call them retroviruses is because their gene expression is backwards. Now, let's think that through, backwards. Well, normally in gene expression, we think about the fact that DNA is converted into RNA, which is converted into protein, right? In a retrovirus, that happens in the opposite direction. These viruses come in with RNA, and the RNA is converted backwards into DNA. What would we call that? What do we call DNA getting converted into RNA? Transcription. So this is called reverse transcription. And in fact, the retroviruses have to come along equipped with an enzyme, a very famous enzyme called reverse transcriptase, that does the job of taking RNA backwards into DNA. And that is why we actually see this virus being named a retrovirus. You'll notice in this cartoon image that there are two strands of positive sense RNA. These strands have to get converted backwards into DNA, and it's at that point that they actually integrate into the host cell chromosome in the form of DNA. So draw your attention, I guess, to these two positive sense RNA strands that are in the capsid of the virus. It is very interesting to take note that for a very, very long time, no one really understood the overall morphology of the HIV virus. The overall morphology was often drawn as icosahedral. That is, the capsid was given that icosahedron shape. But after many, many years of work, there was a group in Oxford in 2006 that actually elucidated that for the most part, the most common form of the virus is actually cylindrical. And that was a breakthrough study. But why is it that it was so hard to just simply determine the shape of this virus? What is it that it does that makes that so hard? Say it again, Jonathan. Okay, good. It's envelope. That is part of it. But also, there's a question on the exam about this. This is one of our shapeshifters. It undergoes a great deal of antigenic variation. Excellent. So we see a lot of shape-shifting, and that's one of the things that made it very hard to determine overall morphology. Notice that on the surface there are two spikes. I think it's actually kind of beautifully ironic that my student that made this model of HIV used dum-dums as the spikes, right? Because nothing could be further from the truth. GP120 and GP41 are, in fact, those surface spikes that recognize very intelligently the receptors on the surface of CD4 plus cells. GP120 binds to them, GP41 mediates membrane fusion. Remember, that is the way that HIV enters, is by fusing its envelope with the membrane of the host cell. Of course, also notice that on the surface, because this is a smooth criminal and it leaves the cell by budding, it does derive its host glycoproteins upon budding, and that is what makes it look very much like self, and you've already drawn our attention to that. Inside the virion, we also see these enzymes that I mentioned that it had to come equipped with. Because it undergoes that backwards process of taking RNA backwards into DNA, we see it coming with the enzyme reverse transcriptase. Reverse transcriptase is crazy. It has multi-functionalities. That is, there are more than one enzyme active site in reverse transcriptase, allowing it to do lots of different things. The virion also comes equipped with integrase. Integrase is the enzyme that allows the DNA, once formed, to integrate into the chromosome of the host cell. That is, to basically splice itself in to the host cell chromosome. Lastly, protease is the last and very famous enzyme that the HIV virion carries with it. We'll look more at what protease does, but suffice it to say, it clips up the long polyprotein that is first formed by the virus. So let's look at what GP120 and GP41 recognize. On our host cells, they recognize those cells that have a CD4 and a CCR5 receptor. Remember how we mentioned that some viruses need to turn two doorknobs in order to get into a host cell? HIV is one of those. Now, who can tell me what kind of cells HIV finds its two doorknobs on? Say it again. Good. T lymphocytes. Excellent. T lymphocytes are one of the types of cells that have those doorknobs. What other types? And certainly we know T lymphocytes to be one of our defender cells. So again, ironically, HIV targets specifically those immune cells. Not just T lymphocytes, but also monocytes and dendritic cells. Dendritic cells are often the first of the host cells that are encountered by the virus because they line mucosal surfaces. So it enters these white blood cells by attaching to these two surface receptors. I like the way that this picture shows that. You can see that the GP120 surface spike, which is the lollipop part of the dum-dum, that's actually binding onto both CCR5 and CD4. CD4 stands for cluster of differentiation, which is a type of an integral membrane protein that's involved in immune function. It actually recognizes MHC-bound complexes. So it's said a very simple way. CD4s play a role in recognizing invasion, ironically, right? That's their role, is in recognizing invasion. CCR5 is a chemokine receptor that hears the voices of invasion, so it too is involved in recognizing when an invasion has taken place. So ironically then, those are exploited by HIV and are where GP120 binds. So I'm going to show you a clip of a video in just a second here. However, I briefly want to mention something that was a bit earth-shattering. Does anybody remember what happened in Berlin in 2007? In fact, it was called the Berlin patient. Well, what happened is, it seems, in fact, that if people are doubly defective in CCR5, okay, that means that they lack CCR5 on their cells. If they're doubly deficient, they do not have it, then what that means is that they do not get HIV. That is crazy, right? In and of itself, that's crazy to think about. Because, you know, HIV is actually flexible, and in later stages in infection, it will not necessarily require CCR5 for entry. It'll use something called CXCR4. And it uses alternative doorknobs, but in the early stages of infection, if it doesn't have CCR5, it can't get in. So this was this knowledge was a bit of a game changer in the case of the Berlin patient, because what happened was he received a bone marrow transplant from a CCR5 doubly deficient donor. So he actually was the first person ever to be cured of HIV. So let's say something important. There is no cure, right, for HIV, but in the case of the Berlin patient, he was cured. And that was because they actually managed to get a bone marrow transplant that was successful from a CCR5 deficient donor. So I want to show you a quick video because I want everyone to understand what happens now. Once HIV binds, what happens as soon as it binds? How does it gain entry? We know it's through membrane fusion. So I want to briefly show you this. And all I'm going to do is play a tiny section of this video just for the visual imagery. So here is the GP120, and there's it binding to the CD4 and the CCR5, okay? Now, this will settle down into those receptors, and you'll notice there's conformational changes. When those conformational changes occur, it enables the GP41 to mediate membrane fusion. So notice that it's that, like, right, the handle on the dum-dum that allows for the fusion of the HIV with the host cell. So if this is our T lymphocyte right here, we can see the envelope on the HIV is about to merge or fuse with that, okay? Yeah, membrane fusion. And notice now that we can get uncoating. Okay, uncoating has taken place. There they are, the two strands of RNA, positive sense RNA, okay? So now the positive sense RNA is inside of the host cell. Let's now take a look at how replication takes place. We now have our positive sense RNA, and I'm going to draw this out. Our positive sense RNA, you can write it as this plus RNA, but what is the polarity of that molecule? 5' to 3'. So let's also draw that out. Reverse transcriptase, as I said, is a multifunctionality enzyme. What its first function is an RNA-dependent DNA polymerase. So remembering, what does DNA polymerase do? It makes more DNA, right? Basically, it builds DNA, right? It does it very rapidly. It builds DNA, okay? Well, there's an RNA-dependent DNA polymerase means that it looks at RNA as the template and uses RNA as the template to build DNA. So we now can draw our 5' to 3' RNA and understand that the RNA-dependent feature of DNA, of the reverse transcriptase, the RNA-dependent DNA polymerase will build DNA. So we will now get, and actually let me draw this down here to stop any kind of confusion. The red is DNA. The blue is RNA. So we now have what's called a heteroduplex. A heteroduplex means one strand is RNA and one strand is DNA. So we can understand now that we have a combined molecule of those two. Next, an active site of reverse transcriptase that is an exonuclease degrades the RNA. So the RNA literally gets chewed up, leaving DNA. We have now a single-stranded DNA molecule, and it's a negative ssDNA. That then serves as the template for the next functionality of DNA, of reverse transcriptase, which is a DNA-dependent DNA polymerase. So this DNA pull takes the single-stranded DNA as template and makes double-stranded DNA. So we now have DNA 5' to 3' and 3' to 5' that now is ready to integrate into the host cell's DNA. So let's just write it can be integrated. And pardon me erasing a little bit of this so that I can fit the word. It can be integrated permanently and randomly into the host cell's DNA by the enzyme integrase. Integrase splices it in. So now with this double-stranded DNA spliced into the chromosome, it is perfectly capable of encoding for viral mRNA. So the viral mRNA can be made, and that viral mRNA can be translated to make polyprotein that is a super long protein. So, sorry, that's bad direction of error. A polyprotein is essentially one massive protein, amino to carboxyl, this long chain, right? Well, in order for this to ever fold into a proper structure of each of the individual HIV proteins, it has to be cut. So protease is the enzyme that comes in and cuts this into fully functional viral proteins. Now bear in mind too that this DNA can of course encode for more plus RNA that can be packaged into daughter viruses. This is the replication of HIV. So if you were hoping to treat this virus, if you were hoping to come up with antiretroviral therapies, what are some of the processes that you would try to inhibit? What would you hope to stop? Go ahead, Sammy. Okay, good. So she says, I would find some chemical that would inhibit reverse transcriptase. And in fact, that was the first thing that people thought of. They actually thought first of something called an NRTI, which is a nucleoside reverse transcriptase inhibitor. Jordan? Yeah, inhibit protease. And this was the second thing that was thought of in trying to target HIV. Protease inhibitors. Good. What else, Nathan? Okay, so the idea then is to stop, you're thinking to stop the synthesis of polyprotein. The hard thing is selective toxicity with that, but okay, Good, good. All right, so integrase inhibitors. This is the most recent drug that has been developed. So these are all lines of antiretroviral therapy. And in a moment, I'm going to write some of them down. Why, even with antiretroviral therapies, why is it still hard to treat HIV? Go ahead. Right. Reverse transcriptase is incredibly error-prone. It does not in any way have the kind of fidelity. Remember, the accuracy that we see in DNA polymerase, the classical DNA polymerase, it actually is incredibly error-prone. So the virus is changing all the time. And while you might think, well, gosh, isn't that a bad thing to be changing all the time? It leads to that antigenic variation that makes it very hard not only for the immune system to recognize it, but it also makes it very hard for antiretrovirals to continue to be effective on the virus, right? So these are all good reasons that it's been very, very difficult to treat HIV. However, antiretroviral therapies have largely extended the life expectancy of those with HIV. In fact, I have an article up here at front that talks about the fact that with good use of antiretroviral therapies, the lifespan of those with HIV has been extended to about 24 years. Now, this is a couple years old, and there are some new antiretroviral therapies. So keep that in mind. That is one positive feature of heart therapy. Let's write all of this down so we don't lose track. I want to make sure this all becomes discussion. We wrote all of this down. So let's write down the fact that unlike DNA polymerase, reverse transcriptase is very error-prone, enabling antigenic variation and quick escape and lack of detection by our immune system. We also know this means they become resistant to antiretroviral therapies very rapidly. Let's just jot down what typical heart therapy is. HART stands for highly active antiretroviral therapy, HART. For the most part, this is not the name that is commonly used for this type of therapy. It has a little bit more of a common name. Who knows what that common name is? The cocktail. So the cocktail is generally a combination of 3 antiretroviral therapies together. Those 3 therapies are often conglomerations of NRTIs and NRTIs protease inhibitors possibly integrase inhibitors, and we didn't talk about it in the context of our last slide, but what else could you inhibit prior to replication? Before the capsid gets into the cell? Membrane fusion. Yeah. So there are also fusion inhibitors. Now, let me explain something a little bit more. Nucleoside reverse transcriptase inhibitors versus non-nucleoside reverse transcriptase inhibitors. NRTIs actually look like nucleotides. That is, they're kind of like base analogs. Remember how those chemicals got in and looked like bases? They were like undercover mutagens. Well, these are undercover nucleosides. They actually get in, they look like building blocks for polymerization, but they aren't. And the most famous one of these is the first antiretroviral therapy that was used called AZT, azithromycin. AZT has on its 2' carbon an azide group instead of a hydroxyl, or sorry, on its 3' carbon, it has an azide group instead of a hydroxyl. So it has, let me say that one more time, an azide group instead of a hydroxyl. What does that mean about its ability to support elongation? It can't, right? It doesn't have an open 3' hydroxyl, so it can't. So that's how that serves to inhibit elongation, and it's a pretty thoughtful therapy, isn't it? Yeah, so these are the cocktail. Now, let's talk about living with HIV. So up on the board, I have put basically the 3 phases of infection. Acute infection happens about 1 to 3 weeks after that first infection, and it is fever, sore throat, muscle aches, lymph node enlargement. You can see why that wouldn't be something that would necessarily call a lot of your attention. Those are symptoms that we get from a lot of things. However, after a little bit of time, that subsides and goes into a very asymptomatic period. That period, depending upon whether antiretroviral therapy is used or not, can be shorter or longer, right? It could be as long as 24 years. During this time, no matter whether you're on antiretroviral therapy or not, the number of CD4 positive cells is constantly decreasing. So you're always seeing that clicking down. Generally, 40 to 80 cells per microliter per year. Of course, that will be less if antiretroviral therapies are being effective. That's why a lot of antiretroviral therapies will actually say on them, this is known to increase your CD4 count. And that's actually several of the articles that I have up here, which I feel like this horrible two-way tear in me every time I look at these articles, because one, these are extending the lifetime of those with HIV, but two, these are highly commercial advertisements. And you look at some of them and you think, oh my gosh, you know, so if I take this particular antiretroviral therapy, will I really be that hot? Right? I mean, it's like sort of, there's this sort of sinking feeling that you have. So I would love to invite anyone who would like to come up and look at these really quickly before you leave. Why are these ads sometimes 3 pages in length? Disclaimers and side effects. One of the worst parts of having HIV and being treated with antiretroviral therapies is the number of side effects that are listed lengthily on each one of these, ranging from nausea, stomach upset, to liver disease, to all manner of side effects. So come on up if you have time, take a look at some of these, and take a minute, if you all would, during your ponderings today, to think a little bit about this topic. Let me stop this.